Thank you very much, Mr. Chairman, for the introduction, and I would like to thank Silvia Misiti and Domenico Salvatore for inviting me here to give a presentation on our work on the role of thyroid hormone signaling in heart failure. That last word didn't make it in the official announcement, but it's essential to the talk. We're going to talk about heart failure. And what I'm going to do is first give a little bit of a background of the work we've been doing that has shown that there's an induction of the thyroid hormone degrading enzyme in heart failure. And then I'll say a few things about some unexpected findings related to this, which tell us something about why this deadenase is induced in the failing heart. And secondly, what additional effects there are of this local hypothyroid condition. But first of all, let's start with the very simple scheme that you've seen a couple of times this morning and this afternoon. What we know, what we think simplified image of how thyroid hormone acts in the cardiac cell as it does in most other cells that we've been talking about. So transported into the cell, we don't know really which transporter is essential, 8 or 10 MCT, but in the cell it binds to its receptor and there it stimulates gene expression. And a couple of the genes that are essential to cardiac function are mentioned here. There's the calcium pump necessary for relaxation. These are two contractile proteins that you will see during this talk. The fast contracting one, which is stimulated by thyroid hormone, was one of the first positively regulated genes identified, and MHC beta, the slow contracting one, which is downregulated by thyroid hormone. Now, as most clinicians will be able to tell you, is that the heart is one of the principal target organs of thyroid hormone. So when we look at some of the characteristics, the heart rate, the ejection fraction, so it's the amount of blood pumped out in each stroke, the cardiac output as a resultant of that, and the relaxation of the heart, these parameters are all sensitive to the thyroid status. Now, in normal life, as Barbara mentioned, T3 is relatively constant, but if there's an increase in T3, you see these parameters go up. The heart rate goes up, ejection fraction goes up, and the cardiac output goes up, and the relaxation goes down. So the heart becomes faster and it pumps more blood. The opposite of the spectrum, hypothyroidism, you see these parameters move in the opposite direction. Now, the other thing that happens if a heart becomes hyperthyroid, so there's more work done by the heart, is that it hypertrophies. And it's the same as in exercise or in pregnancy, when the pregnant woman cardiac size increases by about 30% to compensate for the increased demand, and after birth it goes down again. It's physiological hypertrophy. And cardiomyocytes do not have the capacity to proliferate, as, for instance, liver cells do. When you chop a piece of liver, you get proliferation, differentiation, you got a new piece of liver. This doesn't work in the heart. Cardiomyocytes are terminally differentiated and do not have that capacity to proliferate. At least a very tiny percentage of cells apparently does so, but that's a matter of contention. So now what about heart failure? So for those of you not fully aware of this condition, it's a progressive order of ventricular remodeling. The heart remodels. It wants to cope with the increased hemodynamic load. So this is pressure overload or volume overload. And it does so to reduce wall stress and retain cardiac output. But in heart failure, this initially adaptive response culminates in a clinical syndrome that's characterized by impaired cardiac function. So the heart doesn't contract as forcefully, it becomes slower, and eventually that leads to circulatory congestion and multi-organ failure. And it's one of in some areas, the most prominent reason for hospitalization and a burden on the healthcare system. So it's not a small thing. Now, what causes heart failure? Like I said, pressure overload. This could be pulmonary hypertension, so the right ventricle has to pump against resistance. Aorta stenosis, same thing, but then on the left heart side. Ischemic heart disease, which is a very prominent cause, so poor perfusion, and myocardial infarction as sort of the most extreme case of ischemic heart disease, where part of the heart becomes anemic, no blood circulation, heart attack, tissue dies off, but if people survive with 10–20% of the left ventricle function missing, the remainder of the heart will start to remodel and will start to compensate for the loss of contractile function. But this type of ventricular remodeling is called pathological hypertrophy, because eventually, when the pressure overload or the volume overload is there for long enough, this will deteriorate, the function will deteriorate of the heart, and in this negative spiral eventually results in heart failure, chronic heart failure. Now, if we look at some of the genes that are altered in heart failure, and I draw your attention again to these two guys here, the fast contracting myosin heavy chain and the slow one, then these changes, these reciprocal changes are similar to what you see in hypothyroidism. So this is just one example, there are a couple more, but you see myosin heavy chain alpha go down, as you would see in hypothyroidism, and MHC beta go up, as you would see in hypothyroidism. And that these two proteins explain some of the contractile dysfunction that you see. Not all of them, but there are other genes that move in the same direction. So that gave the idea quite a while ago that the failing heart might be hypothyroid, so to speak, so that the thyroid hormone signaling somehow is impaired in heart failure. And we investigated that. There are several models that we've looked at it as other people have, but I'll just take you briefly through one model of myocardial infarction. So this is a section through a mouse heart, and this is after MI. This is a massive MI. About 40 to 50% of the left ventricle is now scar tissue. Mice can cope with this, and you can get hypertrophy. As you see here, these are the individual cardiomyocytes. You see they're bigger than the ones here in the control animals, and this stuff in between is interstitial fibrosis, which also adds to the dysfunction. But this is the remodeling that you see. But heart function goes down in these mice nonetheless. You see the changes in gene expression and the deterioration of function. Now we measured T3 levels, the idea that this heart is locally hypothyroid, and indeed is reduced by about 50%. And what we found was that this is associated with the induction of this enzyme type 3 deiodinase that you've heard about in earlier talks, the one that inactivates T3 to T2. And this is a fairly rapid induction within the first week after MI, and it persists. So the mouse heart, the failing mouse heart, is indeed hypothyroid and probably related to the induction of type 3 deiodinase. That at least is the simplest explanation. Plasma levels of T3 and T4 do not alter in these animals, so that's not the reason. There's something going on with thyroid hormone metabolism in these hearts. The D3 that we see is indeed in the cardiomyocytes. You can barely make out the individual cardiomyocytes here, and here they are stained for type 3 deiodinase with the antibody that was developed by Domenico Salvatore. And you see in most cardiomyocytes, not all, you see induction of this enzyme. This is a picture that shows there's a higher magnification. Here you see the individual cardiomyocytes, the rod-shaped cells. It's a weird shape, but this is what a cardiomyocyte looks like. And then when you stain it with this antibody or a similar one that was developed by Theo Visser's lab, you see that these cardiomyocytes do indeed express type 3 deiodinase, although some of them don't. Now, we first found this in right ventricular hypertrophy, actually, in a rat model published some 14 years ago. And since then, we have found it in other models, as other people have, in mouse left ventricle, in different triggers, myocardial infarction, aortic stenosis, and also isoporternal, which is a sort of synthetic way of inducing pathological hypertrophy. So the thing is that type 3 deiodinase, at least the way we see it, is induced in the stressed myocardium, so the overloaded cardiomyocyte, leading to this local hypothyroid condition. So in our original scheme, this is basically what it is. D3 goes up, degrades T3, you get a local hypothyroid condition, and thyroid hormone signaling is impaired, so MHC alpha goes down and MHC beta goes up. You see the same thing in human heart failure. Now, so this is the background. So we and others have been trying hard to find out what it is that turns D3 expression on. in these overloaded hearts. And a number of factors, together with Stephen Wang at Children's Hospital, we had a paper on the role of HIF-1 alpha, which seems to be a very prominent regulator of transcription of type 3 deiodinase. But there are other ones, TGF-beta through SMAT signaling, sonic hedgehog through GLI, and MEP kinases. They can all apparently do this when you look at cell culture systems. But none of them have, as yet, been identified as the factor doing this. Now, in a totally unrelated study, we looked at the expression of a family of molecules, microRNAs, and you need to know a little bit about the genomic organization, the piece of the genome in which the type 3 deiodinase sits, and this is called the DLK1 Dio3 genomic region. And it's a very peculiar region because it contains a number of proteins, like Dio3 principally, but also it hosts a lot of microRNAs. Now, microRNAs are small RNA molecules, about 20 nucleotides long. They're about 1,000 or 1,200 identified thus far, and they have a very specific function in fine-tuning gene expression because they can bind to the messenger RNAs, to specific messenger RNAs, and degrade the messenger RNA or reduce translation. So we've only begun to scratch the surface of what these microRNAs do, but it's a very complex new level of regulation. And this particular area is full of these microRNAs. And the activity of this area goes up in stem cells. Pluripotency is associated with upregulation of these microRNAs in this area. And as Barbara also showed in the proliferating precursors of the oligodendrocytes, Dio3 expression goes up. And in some tumors, you see this locus go up. So when we looked at microRNAs after MI, just an unbiased screen, we found, to our surprise, that apart from the proteins that were transcribed, and especially Dio3 going up, what we found earlier, we found that a lot of these microRNAs were actually upregulated. So after MI, when the heart is stressed, we see type 3 deiodinase goes up, and it appears that it goes up as part of this locus being upregulated. And what we think is that, so it's not so much what regulates Dio3 that we have an answer to, but why. It looks like the heart is trying to turn on its proliferative program again. And these microRNAs are not in stem cells or other cells, they are in the cardiomyocytes, we showed that. So it looks like the majority of these cardiomyocytes, feeling the signal of stress, we want to proliferate, they start up this program, which is also present in the fetal heart for that matter. Start up this program, but they can't complete the de-differentiation of these rod-shaped cells, which takes a lot of a cell. Some cells apparently are able to do it, but not in enough numbers. But if this is true, and if we can find why the majority of these cardiomyocytes that turn on this program can't take it to the next level and proliferate, we would have a really good therapy for regenerating the heart. So not dealing with stem cells because there are just too few of them. Now the microRNAs, as I said, it's a whole new field. This is paper 8 years ago. It's constantly expanding the microRNAs that are shown to do something in the heart and are really relevant to cardiac function. So this is nothing to do with the microRNAs I just showed. I'm just showing you this to indicate that these microRNAs are indeed a very viable field of study because they can easily be used as therapeutics. You can give them, you can shoot them up in the bloodstream, they end up in tissue. You can tag them to go to certain places. They're very stable, and you can actually, there are phase 2 trials already going on with some of these microRNAs for certain diseases. So now we know why, we think we know why this locus and Dio3 is turned on, but is there an effect of the local hypothyroid condition? Is it relevant? I mean, some of these genes that I mentioned, our T3 responses, might be affected by thyroid hormone, but, and I'm sorry I have to show you this slide, this is a brief summary, simplification of the pathways in the heart. This is again the outer membrane, the nucleus, the pathways that are involved in pathological hypertrophy and physiological hypertrophy. So for the young students in the audience, if you're thinking about a career in cardiac research, this is what you're up against. But it's at least a lot more interesting than obesity because we know what the cure is: eat healthy and move. So this is where the action is. But it's very complex. But let me simplify it a little bit. These are the pathways that we know are involved in pathological hypertrophy. So here are some of the factors: norepinephrine, angiotensin 1, endothelin. These are all involved in pathological remodeling. If we look at physiological remodeling, thyroid hormone here, we know that it drives physiological hypertrophy, has effects on some genes, and it affects these pathways here that have to do with growth. So it's relatively simple. Now, look, because we knew something about these microRNAs, nothing is known about what T3 does to microRNAs in the heart. So we did a very simple experiment where we took a hypothyroid mouse, so a mouse without thyroid hormone, and treated it very briefly with thyroid hormone. And we looked at the microRNA movement, just to get an idea of whether there is something as a microRNA pool that is responsive to thyroid hormone. So this is, again, a heat map. What this shows is these are 6 individual mice, and hypothyroid ones, and treated with T3. And in blue, these microRNAs are downregulated, and in red, these microRNAs are upregulated. So we found about 56 microRNAs that were upregulated by factor 2 to 16. And what we expected, that most of these microRNAs would be involved in physiological signaling. Well, what it turned out, when we looked at the targets of these microRNAs, so the validated targets, we didn't do this validation ourselves, but this is just in literature, which factors are indeed targeted by these microRNAs. We found that like 56% actually inhibited all these pathways involved in pathological remodeling. And 19% of the targets stimulated physiological hypertrophy. So actually, contrary to what we had expected, it looked like the thyroid hormone suppresses pathological remodeling. It puts a brake on the system. And to put it into that horrible figure, these are the various targets, if you will, of the microRNAs that are upregulated by thyroid hormone. So we think that a consequence of the hypothyroid condition is that it releases the brake that is on the pathological remodeling when T3 is present, so it aggravates sort of the pathological signaling. That's probably also why in thyroid hormone, when hemodynamic load goes up chronically, as it does in aortic stenosis, you get physiological hypertrophy, because in our view, the brake is on the pathological pathways going through microRNAs, so no direct gene regulation. So this is all fine for our studies in mice and in rats, but is there a reason to think that Dio3 might be relevant in human heart failure? And together with a group in Australia who have a huge tissue bank of unused donor hearts and hearts of people who were up for transplant because of stage 4 heart failure. They gave out these tissue microarrays, they're called. They're like hundreds of pores. This is 1 millimeter on one single paraffin slide, and you can challenge them with your antibody. We did these a while ago, actually, these experiments. So this is just one example of a healthy donor, a male, 60 years of age, and this is a person, same age, with ischemic heart disease and heart failure. So this is the worst situation of heart failure because these people are going up for a transplant. And when we analyze a number of these hearts, so the donors, about 75, and the ischemic heart disease ones, you see that there is a quite substantial increase in the expression of the type 3 deiodinase. Activity we have not been able to measure. These samples are a couple of years old. D3 is notoriously difficult to assay the activity, although we can do it in mouse hearts that have been frozen, but we have not been successful with human heart. But we do see messenger RNA going up as well, so we are quite confident that there is more D3. Does it result in lower T3? That's also somewhat more difficult to assess, but here we have the ratio of the T3 concentration in the left ventricle and the right ventricle. So these are the individual patients. And what you see that there is a reduction, about a 50% reduction of the ratio of T3. So T3 seems to be reduced specifically in these left ventricles that are failing, whereas T4 is the same. So to come back to the original diagram, I think with these results we can adjust this picture by saying that T3 in the cardiomyocyte, through its action on the microRNAs, all of this of course has to be substantiated, but the data are quite convincing in our view. Through microRNAs, T3 puts a brake on pathological remodeling. So what happens when type 3 deadenase comes on, you get the hypothyroid condition, which has effects on gene expression like these contractile proteins, but in addition, it releases the brake on pathological signaling. So it would be beneficial to see if we could get T3 in the cardiomyocyte up again. Some clinical studies have been done based on these observations, one from a Danish group last year, but for obvious reasons, these were patients who were not in end-stage heart failure, and they gave a relatively low dose of T3, and it's one of the caveats they put forward themselves in the paper. So they did not see any effect. But my guess is that if this enzyme is indeed there, you would need to hit the cell with a lot more than just a sort of a substitution dose of T3. And then you get into the problem of increasing the heart rate, because that is an effect on the pacemaker cells on the outside. So if this is indeed the case, then we would have to look at something to inhibit D3. Now, if D3 is essential or not, as I speak, mice in Amsterdam, the conditional cardiac-specific D3 knockout mouse is being used for the first time. This mouse was developed here in Naples by Domenico and Monica, and we're lucky enough to have this collaboration with them, so we can actually now test in this mouse model whether knocking out D3 after or just before the MI, so there's no developmental effect of D3 knockout, but just before the MI we knock out D3, and then if our hypothesis is correct, we should see these animals do much worse. So actually this is what I wanted to tell you, so thank you for your attention. I'll be happy to take your questions.